cell biology at the University of Málaga, and also a professor at the Department of Neurology at the University of Texas Health Science Center in Houston. She's a member of Dr. Antonia Gutiérrez CIBERNED group. She received her PhD in cellular molecular biology from the University of Málaga and did her postdoctoral training at the Department of Neurology at the University of Texas, where she has been professor since 2013. Her research interests focus around the study of risk factors for Alzheimer's disease, including diabetes, depression, and traumatic brain injury, as well as the development of therapies for the treatment of neurodegenerative diseases. So her talk today is titled Stem Cell Anti-Inflammatory Therapy to Treat Neurodegenerative Diseases. And with this, Inés, please go ahead. Thank you very much for your willingness to share your work with us today. And you have the floor. So please start sharing your presentation and go ahead.
Thank you so much, Miguel. And thank you for having me here today. So I'm going to share my screen. Give me a second. Here we go. Okay. So as you mentioned today, I'm going to talk about the anti-inflammatory therapy that we are currently developing for different neurodegenerative disorders. So as a start, what I'm going to show you today is the treatment that we have been developing for Alzheimer's disease and Parkinson's. So I'm going to give you a brief introduction about Alzheimer's disease, although we all know about the disorder, but it's one of the main dementia, and is characterized by atrophy in the brain that will especially affect the hippocampal area and the entorhinal cortex, mainly the temporal area. And there is loss of neurons in this area that will finally lead, among many other things, to problems in learning and memory. If we compare this with Parkinson's disease, you can see also that there is also Parkinson's is also a dementia in where the problem or the main problem is that there is a loss of neurons, dopaminergic neurons that are placed in the substantia nigra that will be lost during the development of the disease that finally will lead to a problem with the motor function, but also dementia, as I said. So both diseases, Alzheimer's and Parkinson's, are considered proteinopathies, which means that they both accumulate misfolded proteins in different brain areas. For Alzheimer's disease, we all know that the main pathological feature is the extracellular A-beta aggregation in the form of plaques, but also the intracellular tau in the form of neurofibrillary tangles. And in Parkinson's disease, what we have mainly, there are several others, but alpha-synuclein accumulation in the form of Lewy bodies. There is also an aberrant chronic inflammatory response, including microglial cells and astroglial cells, synaptic loss, and neurodegeneration. And finally, there will be also a loss in the support of neurotrophic factors in the brain. So the main question here is that if we treat neurodegenerative disorders in any way. So just looking at the pathology that we just described, it could be trying to eliminate the misfolded proteins in the brain. We can also try to modulate the inflammation that occurs in the brain. We can try to cure somehow the synaptic loss, or even try to replace those neurons that are lost, or even replace those neurotrophic factors that are decreasing during the development of the disease. So one of the approaches that we took to try to find a cure or something to ameliorate the problems that occurs in Alzheimer's and Parkinson's disease is the use of stem cell therapy. So there are no disease-modifying therapies for these neurodegenerative disorders, or other, there are some pharmacological approaches that can ameliorate this disorder, but there is not really a cure. So as I said, one of the things that we can try to do is to develop a cell therapy using stem cells. There is more development in the Parkinson's area, in where there are already several approaches trying to do intracerebral graft in the brain of the human patients. There are for sure several pros and cons. The cells in the graft need to survive within an adult brain, which is something really complicated, but some of them can survive. Depending on where the graft is produced, then cells need to migrate to the target region. For sure, the substantia nigra is an area that is difficult to reach when doing the graft. They need to get differentiated into dopaminergic neurons. They also should, if you want to produce new connections, they need to make those connections with the surrounded neurons. And also with time, it has been observed that the cells that are in the graft will also get those misfolded proteins, because there is a cell-to-cell transmission from the diseased brain to the grafted cells. And for sure, this is a very invasive method. So on the other hand, the other way that we can try to put those cells into the brain of these deceased patients is doing peripheral transmission administration. They don't really, the cells that we are administrating, they don't really need to survive for a long time, because we can do periodical inoculations, so they don't need to last for a long period of time. They don't really need to migrate through the parenchyma of the brain, because they will go with the blood irrigation that there is in the brain already, hoping to reach the target area. They don't really need to differentiate, thinking on that they also will produce different factors by themselves. So if the target is the production of different neurotrophic factors, then there is no need for synaptic connection, and for sure is much less invasive than the graft. So this is our working hypothesis. What we are trying to do is to inoculate intravenously, in this case, neural precursors, and that also can release different factors that can be used as a novel, non-invasive therapy for Parkinson's and also Alzheimer's disease. So the main goal is to develop this approach targeting these two different disorders, to have an effective and low risk, even personalized, because I'll show you that we can do this also using induced pluripotent stem cells, trying to reduce the pathology in these patients. So the first thing that I'm going to show you is how we investigate how this peripheral administration of stem cells will produce in Parkinson, Alzheimer's animal models, with this in a preclinical model. And then also we evaluated the potential side effects that this therapy can have. So first, I'll talk about the Parkinson's. This was done by George Edwards. He was one of my PhD students that already graduated, and also was continued by Nazareth Games, that is currently finishing her PhD. So what we did was to have two different groups. In one, we analyzed the toxicity of the inoculation of these neural precursors. We had the control with just the vehicle, and then we sacrificed the animals at different times, especially looking at 180 days after inoculation for the potential formation of tumors. And we also did this therapeutic group, in where we use for this the Parkinson's model induced with MPTP. I'll show you later how this works in case you are not aware about. And we injected neural precursors that were derived from mesenchymal stem cells or embryonic stem cells, for sure, from mice. We have our controls. We injected the animals twice, each time with 1 million cells, and then we analyzed the animals either 10 days after the inoculation of the cells or 3 months to see a long lasting effect of this therapy. So this is the generation and the characterization of the neural precursors. These are the embryonic stem cells, and these are the neural precursors obtained from these embryonic stem cells. We also did the experiments using, as I said, mesenchymal stem cells, derived neural precursors. You can see here the morphology with the specific markers for the neural precursors, and we specifically selected this because this is a neural lineage, so they potentially can convert or derive into neurons and astrocytes. Genetically, they are more stable, more than a stem cell that is not so differentiated. Still, they have limited propagation, which is okay. They have no ethical concerns compared with, for example, the embryonic stem cells. Potentially, they lack of tumorigenesis, I hate this word, for capacity to form tumors, and then these mesenchymal stem cells that cells can be obtained from patients, from their own cells, just thinking on the application, the future application of this therapy. So the first thing we did was to analyze these animals just in case they were able to produce any type of tumor. You can see here we analyzed the lung, spleen, kidney, liver, some of the organs where probably they can travel and stay there and probably form tumors. We couldn't see anything in all the animals that we analyzed. We also did the staining with Ki-67 to see a high proliferation rate in some of the tissue. We couldn't see anything, so we really believe that this is a safe therapy. For the therapeutic model, we use the MPTP animals. MPTP is a drug-induced model in where we inject the MPTP, that is a drug, intraperitoneally, and it will be able to reach the brain and attack specifically to neurons that contain the transporter for dopamine, and will selectively kill dopaminergic neurons, specifically in the substantia nigra, as you can see here in the pictures, in where we stain with T8 neurons, and you can see how the number of these neurons are decreased. So the animals, you can see that they have problems with the motor control, and they have this neurodegeneration in dopaminergic neurons. So we injected these animals, as I said before, and then we assessed different behavioral tests. And for example, the rot for the motor coordination, including the wire hang, also open field for velocity and the gait of the animals and the exploratory features. And then we also perform nesting that is also depending on the dopaminergic response. So you can see here after 10 days post-treatment that the animals here you see in the white column are MPTP animals injected with PBS. Then in gray you have the MPTP animals injected with neural precursors obtained from mesenchymal stem cells. In black you have the neural precursors obtained from embryonic stem cells, and then this line column you can see animals that were not injected with MPTP, meaning that they are somehow the wild type or the control for the disease, just injected with PBS, also for the control of the manipulation and the injection. So you can see here that just injecting the cells, we see an amelioration in in the strength and the ability to cross this wire with the animals injected with both types of neural precursors. Similar results we obtained with the rotator, with their ability to walk over the road when was accelerating. And with the time, after 3 months post treatment, we were able to see something similar, but more reduced, thinking that the for sure the benefits that they could have after the administration of the cells was reduced over time. And then you also need to remember that these MPTP-induced animals are not a model for progression of the disease, since it's just one single injection, and then it does not progress as it could happen with transgenic models for the disease. We were wondering if this was due to cell replacement. No. We did staining of tyrosine hydroxylase for dopaminergic neurons. We look at the substantia nigra, we look at the striatum and where they project, and we could not see any difference. So we are not really replacing or making even the neurons to produce more dopamine, but we were doing something because we could see some amelioration in the behavior of the animals. So what we did then was to look at the inflammation. We may be, in fact, modulating the inflammatory response. So we went and looked at microglial cells and then activated glial cells with the CD45. And you can see here, these are the controls. I don't know if you can see my mouse. You have on the top the control, and then in the other ones, the animals injected with neural precursors. If we quantify this, we see here the control and the injected animals. We have a decrease in the amount of cells that are stained for IVA1, microglial cells. And then if we look at specifically activated microglia with CD45, we also see a decrease, reaching levels very similar to non-MPTP injected animals. If we look at the astroglial cells by DFIP and also BLBP for unreactive astrocytes, we see something similar. If we do quantification in control, PBS injected animals and neural precursor injected animals, we see the same trend of a significant reduction in the amount of GFAP immunoreactivity in treated animals compared with control animals. So what we could see from this Parkinson's therapy is that we were trying to first do a cell replacement. Originally, that's what we had in mind. But the cells need to be delivered in the blood vessels and then cross the blood-brain barrier and then get into the brain and do all the things they need to do. We have been looking for those cells in the animals. It was kind of difficult to find them, so we don't think that they really last for a long time. But for sure, what we are not doing is replacing the cells that are already lost. So what we probably are doing is an immune modulation. Probably cells, when they are injected, they are secreting trophic factors and growth molecules that may be modulating what is already there. They may also be producing some antioxidative stress substances that we don't know yet. Or may, in fact, favor or benefit the synaptic plasticity or even the neural function of the neurons in the brain. So next, Nazareth continue with these experiments in animals for Alzheimer's disease. We did both using animals for APP for accumulation of amyloid beta and also the B301S for accumulation of phosphorylated tau. And we did two different groups. We treat treated the animals early on when they were 3 months old, before they start having the accumulation of A beta or even the phosphorylation of tau, and then sacrifice the animals by 7 months. And then we also have a late treated group when they already have this pathology in the brain. So we injected the animals at 6 months and sacrificed also by 7. You may already know about these animals. Just to tell you that the APPPS1 and the P301S animals, they both start having this accumulation in the brain by 6 months. Again, in this case, we injected neural precursors, both coming from mesenchymal stem cells again, but this time we try with induced pluripotent stem cells and then derived two neural precursors. The idea was that this could be used in the future for the treatment of humans, therefore getting fibroblasts deriving into inducible pluripotent stem cells and then having the neural precursors. And this is what we did, but with the mouse cells. Again, we analyze different organs after a long time just to see if they were able to produce any type of tumor. Nothing was seen, any type of anatomical alteration. And then we perform the cognitive assessment. In this case, we did Barnes maze and reversal learning for these animals in the early treated group and late treated. And you can see here that this is the learning curve for the injected animals. And you can see here that with 7 months, the APPPS1 animals injected with PBS are not able really to learn anything as the learning curve is almost flat. But then you see the animals injected with neural precursors that they to learn and they take less time to reach the platform or the escape box in this case. Similar thing happened with the late treated animals. They also learn much better and faster than the PBS injected animals. When we analyze this in the tau animals, we see some of the groups ameliorating the cognitive impairment. As you can see here in the early group, what we observe is that just the animals injected with neural precursors that were obtained from the mesenchymal stem cells was performing better. The opposite happened with the late treated group in where animals injected with neural precursors coming from inducible pluripotent stem cells perform better than the other. As the tau animals also have motor impairment due to the accumulation of this hyperphosphorylated tau in some brain areas, we also did rotarod, and we also can see in the early treated animals and late treated that when we inoculate neural precursors into these animals, we can see that they perform better also for motor assessment, meaning that it also ameliorates the impairment they have in motor coordination. We then move to the inflammatory response. As we saw before in the Parkinson's, that we were modulating a little bit or substantially in inflammation. We look at the same in these animals. Here you can see that we do see a little bit less DFAP positive astrocytes. It's not as much as we expected. If you see the quantification, if we see the amount of astrocytes around the thioflavin positive plaques, it looks to be less than in the PBS treated animals, but it's not as strong as we saw before. We check for different cytokines. I'm showing you just some of them. For example, we see that inoculation, this is late treated animals, inoculation of neural precursors are able to increase some anti-inflammatory cytokines and decrease some pro-inflammatory cytokines and interleukin-1 beta. Something similar happens with the tau animals, but it's not as strong as what we see in the APP animals. So we see a little bit of increase in anti-inflammatory, decrease in interferon gamma, but we can see, in fact, a little bit of increase in TNF-alpha in these treated animals. So what we have till now is that we have been able to analyze a new therapy in a preclinical model for different neurodegenerative disorders. We are using neural precursors that can be obtained from embryonic, mesenchymal, and induced pluripotent stem cells. The good news is that they are able to ameliorate clinical signs that we can observe in animals, both motor impairment and cognitive impairment. We realized that we are not doing any cell replacement or we are neither reducing the deposition of amyloid proteins. I haven't shown you, but we can't see a reduction in amyloid beta deposition or tau phosphorylation. What we can do is to modulate somehow the inflammatory response, and we are currently on the way of developing this therapy, but cell-free. So we are analyzing the neurotrophic factors that are released by these cells to see if we can try to mimic exactly the same, but without the cells, just the factors that they are producing. So I would like to thank my previous group in Houston, my current group here in Malaga, and for sure I couldn't do anything that I show you today without the help of Dr. Antonia Gutiérrez and her group. Thank you so much.
All right, thank you very much, Inés, for a very nice presentation. So the paper is now open for questions. So again, I remind you that if you're interested in making any questions, please use the chat option that you have on the upper side of your screen, and just send me a message stating that you're interested in making a questions, and I will give the floor to anyone interested in a first come, first served basis. So I will wait for a few seconds. There is a question from Ignacio Torres Alemán. So Ignacio, if you want to just put your microphone on and the camera and go ahead, please.
Yes, thank you. Maybe I missed the detail, but where is the site that you place the injection of the cells in the Alzheimer's model?
No, I think I haven't said that. In the vein of the tail.
Okay. Well, this is important. Thank you.
All right. Thank you both. Any other questions? While we wait for additional questions, let me just remind you that you can access to the whole list of future webinars in the CIBERNED webpage. So we will plan to have a webinar every other week, always on Thursdays at 3:30 p.m. And since this year the webinars will be open to everyone, anyone interested. So please feel free to either resend the link to the presentation or to just spread out the word of the webinar series, and everyone is more than welcome to attend. So we have another question from Sara Hernando. So Sara, please put your microphone on and your camera on as well, and go ahead.
Okay, I think that you can see me. I don't know if you see me properly.
Yes.
Okay, fine. So I would like to ask where do you measure the cytokines for the AT animal model? In the serum or in the brain?
In the brain. In the brain, we did brain homogeneity, we obtained the soluble fraction, and then we measured in the soluble fraction.
Okay, no cerebrospinal fluid, just brain.
No, just brain. Yeah.
Okay, fine. Thank you.
All right, thank you both. I think I have a raised hand here. Let me see who is. Okay, Javier Sáez Valero. Javier, go ahead, please.
Hi, very nice talk. Thank you. And I don't know if I missed the detail. I think that you say that you injected 10 million of cells.
Yes.
Which is regarding and you already see an effect, you already see increase in neurofactors, but which is your do you think that for the translation to human, do you need to increase to a very high number of cells in order to see any effect? I know that is far away, the translation to human, but I would like to know your opinion. Thank you.
Yeah, that's a great question. So the idea was to inject more cells. At the beginning, we tried to inject more cells. If when we tried to do that, then we were using the jugular vein because you have a higher diameter, so you can inject more. But they had problems with the injection of many cells. They got the lungs were clogged easily, and they died. That's why we moved far away from the lungs and injected in the tail vein. So we were injecting 2 million because that's the range in where it was a solution that it was not much volume with enough cells. So everything was, we were trying to standardize everything. The goal for human translation of this work is not really injecting cells. So we are now moving to the area in where we are analyzing the condition media in where cells are growing to try to inject what they produce, but not the cells. How much, how many injections, for how long, that we don't know. We still don't know, in fact, how long they can survive in the animals. We are tracking the cells. We think they are able to survive between 1 and 3 days, depending on the quality of the cells that we are injecting. And we do know they reach the brain. But how long they stay there, then that's difficult to know. Or if they, in fact, they differentiate into something different that is not the neural precursor, but is finally glial cells that is derived from the inoculum that we injected.
Thank you.
All right, thank you both. So we have one other question from Laura Herrero. Laura, please go ahead.
Hello, Inés. Thank you so much. I really like your talk and very interesting results. So I was interested about the viability of the mesenchymal stem cells and neuroprecursors when you inject them. So for how long do they live inside? And also, what is the percentage of the cells that really reach the brain, or do they go also to other tissues? Because I thought the tail vein injection is very interesting, very easy, also practical, but then how do you know it doesn't go to other places?
Yeah, so that's a great question. We have done many things to try to see what how many cells are staying in the brain and for how long, right? So we did live imaging, but the amount of cells is so little that it's so difficult to track really in live imaging how many they are and where they go. If we do, they are labeled with GFP, so we can somehow track them. We can find them 1 day after the injection, 24 hours after the injection. They are mainly in the brain. That's a good thing. They are mainly in the brain, but you can also find them in the liver. Probably we are detecting something that is already degraded or something like that. Then for how long? For sure, 24 hours. We have done 1 week. We can't see anything. So guessing between 24 hours and 1 week. No, we haven't done all the times. We haven't done like the kinetics over the time, but that's something that is pending. I don't know if that will really provide us much more information, since we are moving away from injecting the cells and more injecting what they produce.
Yes, and this goes to my second question and the last one. So maybe it will be interesting to do like a gene therapy. So use these cells to target, maybe, as you were saying, right? Maybe something that they produce. So maybe you could do the gene therapy with lentivirus, for example, for these mesenchymal stem cells, and then use an anti-inflammatory protein that once it reaches the brain, it will help to have stronger results.
Yeah, so what we have done is to analyze the culture media where they grow, and what we can find really is a huge amount of BDNF and TGF beta. And that's something that it has already been reported, that there are some of decrease in the production of BDNF in, for example, Alzheimer's patients. So we are kind of matching what is already known, but for sure they should produce something else that should be more than just the BDNF. So yeah, I mean, I think that your way of continue with this investigation, I think that that's also something to keep in mind. Thank you.
Thank you, Inés, and good luck.
Thank you.
Thank you both. So we still have some time for additional questions, if there are any. I don't think I've received any other question, but we can wait for a minute or two to see whether someone else want to make any questions. I remind you that if that's the case, please send a message through the chat, just stating your name. No need to write down the question, and I will give you the floor. So I don't see any raised hand or any other questions through the chat. So that's your last chance. If you want to make a question, do it now. No questions? All right, if so, I think that's, we're gonna close the session here now. Not before just thanking Inés once again for his willingness to share with all of us her work, very interesting. And also everyone for attending the webinar. And I remind you that we will be back with the next seminar in 2 weeks on Thursdays, 3:30 p.m. as usual. So thank you, Inés. And thank you so everyone. Much. Have a great day. Bye-bye.
Thank you.